tell you a little bit about this wonderful scientist. Dr. Dzirasa was the first African-American to complete his PhD in neurobiology at Duke University. And he, in addition to his PhD, he completed his MD at Duke in School of Medicine in 2009, and his residency in general psychiatry in 2016. And his research interest focuses on understanding how changes in the brain produce neurological and mental illness. And Dr. Dzirasa work has been featured in different news venues, including CBS 60 Minutes, and he has been awarded several prizes, including the International Mental Health Research Organization Rising Star Award, the Sidney Bayer Prize for Schizophrenia Research, and in 2017, he was recognized as 40 under 40 in health by the National Minority Quality Forum and the Engineering Alumni of the Year from UMBC. He was also awarded the Presidential Early Career Award for Scientists and Engineers, which is the nation's highest award for scientists in the early stages of their independent research career. And that was in 2016. Last week, we had Damien Fair, who also had received this presidential award. So we are in good company. And in 2019, Dr. Dzirasa was awarded the Alan Leshner Public Engagement Fellowship from the American Association for the Advancement of Science and the Society for Neuroscience Young Investigator Award, given to the best young brain scientists in our community. And has served as Associate Scientific Advisor for the journal Science Translational Medicine. And he was a member of the 17th Next Generation Research Initiative mandate by Congress. And in addition, he has served in the editorial advisory board for TEDMED and the advisory committee for NIH Director for the Brain Initiative. And at the present time, Dr. Dzirasa is the Ranga Krishnan Associate Professor at Duke University with appointments in the Department of Psychiatry, Behavioral Science, Neurobiology, Biomedical Engineering, and Neurosurgery. So, Kafui, it's such a pleasure to have you, and we welcome you to Puerto Rico and to our BPN Doer Seminar Series. So, thanks, and we look forward to your talk.
Well, thanks so much for having me here. The good news is I am repping your gear. Thanks so much for getting it over to me on time. So even though I'm not physically with you, I am totally with you in spirit. The bad news is, unlike your speaker from last week, I am not a genius. and the real bad news for me is I'm no longer under 40, as all of my grad students and postdocs point out. All right, let me go ahead and screen share. All right. And I'm going to assume that if everyone can't see that, someone will come on and yell at me.
Yeah, we can see it. No problem.
All right. So this is me. And I've always been an engineer, right? Whether it was like fixing bikes with my siblings on the weekend, or I don't know what that is in my hand. I'd like to think it's a tennis ball. Maybe it's like an apple, me trying to like fake like I'm Newton or something. But I've always thought about how things work together in a system and how you take them apart and pull them back together. It's who I am at my core. And it's fascinating. I have a degree in neurobiology, and I always joke that the only reason they consider me a biologist is because I happen to get the things I analyze out of animals. But otherwise, my full construction is an engineer. I was an engineer in undergrad, chemical engineering major, and then finished up and wanted to just figure out how to take the engineering skills that I had and use them to help people, right? It's sort of like this broad idea. I'd spend a bunch of summers in undergrad working in all kinds of different sectors. I spent a summer working in an environmental toxicology lab, and then I went to Mobil technology company. This was before Mobil and Exxon merged. And I was working on creating mathematical models of the process that they use to make gasoline and refine it. And, you know, at the time, I'll say, you know, gas was like 90 cents a gallon. And I couldn't imagine that like working on environmental toxicology or like gasoline could have anything to do with helping people in the world. You know, fast forward 25 years later and we have global climate change. But at the time, I just wanted to figure out how to use engineering for something that would change people's lives. And near the end of undergrad, I decided that what I really wanted to do was take my engineering, fuse it with an understanding of basic biology, and then do something in the medical space to help people who are suffering. And the area that I got really interested in was brain-machine interface. And I think you can appreciate quite quickly how it's an intersection of these three, right? So the cool technology at the time was people had figured out how to read electrical signals from the brain and then send those to a computer, which could decode those electrical signals and then use them to move robotic parts, right? So you could basically think and a robotic prosthetic would move. And this area was called brain-machine interface. So this was awesome for me. It was like all engineering, all neuroscience. And, you know, if somebody had an injury to the spine or lost a limb, you could basically recover it, right? It was like Star Wars meets engineering meets biology meets medicine. And when I got to medical school, after my first year, you do all your second year of rotations in the clinic. And this is at Duke University. And it was the first time I encountered someone, or clearly encountered someone with mental illness. I was at the State Psychiatric Hospital. And I'll never forget the experience. It was the first time I directly had tried to interview somebody with schizophrenia. And I remember for the first time thinking, oh my gosh, mental illness is a real thing. And you suddenly, I grew up outside of a city, so you suddenly flash by all the people you walked by that were like homeless or talking to themselves. And you realize like the whole time they had a serious psychiatric illness. And what was so unique for me in thinking about psychiatric illnesses were here are these illnesses that were clearly attached to the same organ that the neuroprosthetics, the brain-machine interface on the left, was working off of, but nobody could tell me how the organ changed, right? And how the biology of the organ changed. And I thought to myself, wow, if we can create brain-machine interface devices for psychiatric illness, we'd really be able to help a lot of people. So that's what I'm gonna do. I'm gonna create brain-machine interface devices for psychiatric illness. But one of the things you could appreciate when one starts talking about psychiatric illness and emotional health, what you realize is that the key piece of psychiatry and understanding emotions is what we call the self-report. In other words, you know how someone is feeling because they tell you how they're feeling, because you ask them. And so that became a really hard challenge for me to figure out how to solve as I was thinking about brain-machine interface. How do you get at the biology without somebody telling you what they're feeling? This is a tool called functional magnetic resonance imaging, or fMRI. And what fMRI does is it basically cheats. So it says when brain tissue becomes active, it starts ripping oxygen off of hemoglobin, right? So as you need more, as your brain becomes activated, you use more ATP, and then you need oxygen, right? And so you can look at how much oxygen is on the hemoglobin as a proxy of how much brain tissue is activated, so chunks of brain tissue. So what they're doing in this case is that they're basically saying, let's look at a whole brain, and we're gonna ask people what they're feeling, right? So it's a series of experiments that they've done, but at the last step, they basically ask what people are feeling, and while they're looking at their whole brain. And there are a couple of important things that come out of this fMRI, this functional magnetic resonance imaging study, right? So you get the self-report, but then you can map the self-report onto what you see in the brain. And the first observation, which is a really important one, is that emotions, here the fMRI is giving a picture of the brain like every second or so. So emotions are these things that are showing up on the brain, and they're there over seconds of time. And this is really important because a lot of what you think about brain-machine interface for like neuroprosthetics and moving your fingers, you want the processes to be really, really fast. But emotions are constructed in a way that evolves over a much slower period of time. It's why you feel happy for a second, or you feel angry for 2 or 3 seconds, because they're much slower. The second thing that's equally as important is these, and you can see this from the pictures, the emotional states are what we call distributed. In other words, you don't just have a small chunk in one area that's happy and a small chunk in another one that's sad. You have brain areas, multiple brain areas that coordinate together to produce the emotions. So this was sort of the framework that I had going into grad school. Now, interestingly enough, much of what was going on in the field of neurobiology at the time when I got into grad school was not actually looking at the whole distributed system, it was actually just zooming in. So what happens with zooming in is you're basically interested in what the individual cells are doing. And so I spent a lot of time in grad school learning how to zoom in. And so by zooming in, we would record electricity from cells. And then we could do even more complicated things. We can put many, many wires down. So instead of one wire to record one cell, we put many wires down and record many cells, but we're zooming into a brain region. I'm just showing you a plot on the bottom of a series of cells, about 20 cells firing, 20 to 30 cells firing across time in a part of the brain that controls motor behavior, right? So we're just zooming in an area, and this is much what was going on in the area of neuroscience at the time. And you can see all the cool stuff that can be done with zooming in, right? You can record brain activity and then record movements. I'm just showing you some cool work where they're looking at a monkey moving a joystick or moving its arm while you're zooming into an area of the brain. And then you could just map the electrical activity in the brain area to the movement of the arm. So this is the type of work that was going in on the time. People were zooming in into the brain to understand how the cells related to behavior. Now, you can appreciate how much can be lost when you just zoom in. So these are a depiction of water molecules, right? You zoom in to water close enough. It doesn't exactly look like this, but it's an artistic description of it. And you have a series of water molecules on the left and on the right, and they look identical because it's the exact same picture. But when you zoom out, you can realize something really different is going on, right? And when you zoom out on the left, you see a really tranquil beachfront. And when you zoom out on the right, you see a tsunami. And the reason why this is so important, there are even emotional experiences that you can get from seeing these pictures that you wouldn't appreciate if you zoomed too far in. So it made me think a lot about this idea of, while much of the field was zooming in and deeper, how would I succeed in zooming out in order to see what was the distributed state of emotions in the brain? So I started recording electrical activity from many sites in the brain. So I put electrodes in many sites of the brain simultaneously in a mouse. You could see a picture on the bottom right in which the blue sort of things going in the animal's heads, those are electrodes. We've just taken a picture of them using a CT scan. And you can see that these wires end up in many brain areas. And so I'm recording activity, zooming out and recording brain activity from many brain regions simultaneously. And because I had a background in engineering, it was fairly simple to figure out what to do with them at first, right? So for each brain wave that we're recording, these are the same sort of brain waves that you get from the scalp when you're looking at sleep or epilepsy signals, except we're recording these from inside the brain, those blue wires. And we could take these brain waves and we can filter them across different frequencies and ask how much activity was in each of these frequencies that made up the wave. The other thing we could do is we can quantify how much brain waves change together across time. The engineering principle here is things that change together across time tend to lie within the same system. So I'm just showing you how the red peaks are lining up here. This is called coherence, or these two brain waves tend to change together, cohere, or synchronize across time. We can also do a lot of offline mathematical analysis. We can shift one wave relative to the other one and ask if that synchrony or coupling gets better, or we can shift in the other direction. And if we say the coupling is better when area 1 precedes area 2, then we can infer that information is moving in that way in the brain. And if it gets coupling gets better the other direction, then we can infer that information is moving in the other area. So now I zoomed out. I was recording electrical activity in many, many parts of the brain. I was seeing what was going on in brain areas and how information was synchronizing between brain areas. And suddenly the data was far too big than for me to know how to analyze. There's just it's like 10 mice and 10,000 bits of data from each mouse. So it's just way too much information. And the way I ultimately ended up solving this is I zoomed out even more. So it turned out that this collaborator here, Lawrence Karen, who was over the computer and electrical engineering department at Duke, he is in the area of machine learning or artificial intelligence. And what he does is he takes lots of data and he tries to find patterns in lots of data. So it turns out the physics of the upper atmosphere mirrors some of the physics that you see when you zoom out in the brain as well. So I basically went over to somebody who thought a lot about a field called geothermal imaging. In other words, looking at thermal patterns in the atmosphere on the planet. And I asked if he would be interested in working together to see if we can make sense of the massive amount of data we were recording when we zoomed out of the mouse's brain. All right. And I'm going to walk you through this fairly quickly. I'm going to orient you this, but I won't spend a whole lot of time here. This is what a machine learning model looks like. So this is how we make sense of all that data that we're grabbing in. And I'll walk you through this systematically. What we're essentially trying to do, and I think the best way to think about this is trying to understand music, right? So we start off with the music. And like when you hear music, it is basically really fast waves. The human auditory processing is about 10,000 times per second. So it's really fast waves that are striking your eardrum, right? And so we'll say the LFPs, or the local field potential waves, are like the music, right? So we're going to take the music, and then we're going to chunk it up into 1 second bins, like we chunk up the LFP. So 1 second bins of music. And then what we're going to do is we're going to extract out of the music, we're going to extract a series of tones. And those series of tones will be like the things I told you before. In other words, the brain waves in each frequency, and then the information synchronizing, or then the things leading or lagging. So we can essentially chunk the tones out of the music. And then we're going to use a set of tools, basically a neural network or artificial intelligence, to say, well, what's the pattern of tones that tend to be changing together? And that essentially gives us our chords, right? So tones that change together are the chords and the instruments playing. And so we're building a statistical model that takes music, it finds the tones, and then figures out what the chords are that make up the tones. Then the next thing we want to do is say, well, how does the chords that are being played relate to the emotion that's being reported? And I'll show you how we report emotion out of a mouse. But part of the model is going to relate the chords to the emotion. And then finally, we're going to say, well, now that we've learned the motion and the tones and the notes, how do we make sure that when somebody listens to this, and it might be a different genre, we still know it's the same sort of music, right? So you can listen to a song in a bunch of different genres, and you still know it's the same song. So that is what the machine learning model is essentially built to do. All right, and so I'm going to show you how the engineering and basic biology and the medicine came together to achieve this. This is, as I mentioned before, I went to medical school, and so I think a lot about other fields of biological function and the success that they've made. So on the right, you can essentially see, imagine a clinical case in which someone has a heart attack or heart failure. And what you ultimately want to do is make the heart pump more normally. And we have medicines that do that. Those medicines can be engineered medicines like a pacemaker. It could be ACE inhibitors or digoxin, which they used to use when I was in medical school, to make the heart contract more normally. But what you appreciate if you look at the left is that, young people tend not to have heart attacks. And so one of the biggest risk factors for having a heart attack is aging. And so if you study people across time, and physicians have done this, it turns out you can determine pathways, biological pathways in young people that is related to the risk of having a heart attack in the future. These are things like blood pressure and cholesterol. And so once, our cardiology friends realize this, they could start developing treatments that lower your blood pressure or lower your cholesterol as a way of preventing the heart attack happening on the other side. And as you can clearly appreciate, the best treatment for the blood pressure or the cholesterol problem isn't the pacemaker. You don't necessarily want to use that up front. There are other biological pathways that you could target early on. So I wanted to know if the framework like this could be used for something like depression, right? So we can essentially take individuals who have major depressive disorder, and we can give them antidepressants. And the goal of the antidepressant is to make their brain function as if they do not have problems with mood or eating or sleep. But like the heart disease case that has aging as a risk factor, severe stress is a huge risk factor for developing mental illness as well. So I wanted to ask the same question. Could we essentially go before the major depressive disorder is developed and find some sort of vulnerability signature that looks a lot like blood pressure, cholesterol, but some sort of vulnerability signature that determines who will get major depressive disorder if they are stressed? So here's how we do this. We could take our model organism, in this case is a mouse, and then we're going to look at a whole bunch of brain areas that are involved in processing reward and social processing and sleep. And we're going to zoom out and we're going to put electrodes in a bunch of these brain areas that you see in red. And then we're going to record activity from those brain areas. And then we're going to take our mice and we're going to put them through a lot of stress. And when you put these mice through stress, about 40% of them look just fine. In other words, we call them resilient. And about 60% of them show a phenotype that looks a lot like depression. They don't sleep well, they don't like food anymore. And so we're just going to ask, is the music in their brain different before you put them in stress? And does that music tell you which ones will ultimately be susceptible to stress or resilient to stress? And this is what the music looks like. Just imagine this is like, your music sheets. It tells you what's going on in each brain area. And you can see that along the diagonal. At the off diagonals, you see what's going on between brain areas. In other words, are they playing chords together? And then we also know for each time two notes are playing together, two instruments are playing together, we also know who is leading and who's lagging. In other words, who's the conductor and who's following. And so we get this huge network that tells you about all these brain areas, but now the information is all synthesized together into a chord. And we could see and we can watch that chord playing across time. So when this chord is being played in the animal's head, the animal is much more susceptible to future stress. In other words, it's a vulnerability signature. And then what we're able to do in a series of studies published in 2018, we're able to show that a bunch of manipulations that cause or give risk for depression in humans also make the signature show up. So we showed that a molecular manipulation, in other words, a single molecular pathway could make the signature show up. If we gave a drug that when you use it to treat liver toxicity in humans from hepatitis C, this makes individuals, humans, feel depressed or suicidal. This makes the same signature show up in our animals. If you put our animals through childhood trauma, it makes a signature show up. And then finally, this signature is related to sex differences as well. So in other words, this is a robust vulnerability signature that holds up under all these conditions. So what we're doing in my lab now is that we're doing a series of experiments to find other network states or other emotional states, things that are linked to social reward, anxiety, aggression, feeling negative, and anticipating food reward. And the idea now is that we can have objective measures of these emotional states in animals' brains, not by zooming in, but by zooming out. Now, I don't want to take any credit away from the power of zooming in. Much of neuroscience is moving in a beautiful, brilliant direction of zooming in. We've got all kinds of new probes now. So imagine in the picture I showed you when I was doing in grad school, it was one probe. This single probe that's about the same size now has about 100 contacts. So you can record 100 times as many cells with that single contact as I could when I was in graduate school. And so you could see the power in this case of zooming in. You can record many, many more cells now. There are other techniques for zooming in as well. This is literally using a microscope to zoom in and looking at the activity of cells by seeing the calcium in the cells change over time as the cell is firing. And I think there's a lot of critical importance for these techniques and technologies that are being used to zoom in. But one of the challenges we have in psychiatry is figuring out, well, why do you respond a different way when you see the same thing? Right? In other, why does the car driving by or the TV show or a person talking to you, why do you respond differently when the stimulus is the same? And the way we deal with this in neuroscience labs is we exert an incredible amount of experimental control, right? We take away all the experimental variables such that the response is quite consistent. But this would never work in a human case when you're trying to understand psychiatric illness, right? The mapping between stimulus and multiple output responses is really complicated to understand if you zoom in. But if you zoom out, we think there's a framework that becomes a lot more clear, right? So if you zoom out, you can appreciate that the brain is actually entering different emotional states. And then if you give a stimulus, the response is based on the state that the brain is in. So we think if we ultimately combine this idea of the tools that you can use to zoom in with a framework that you can only identify by zooming out, we'll be able to better understand how the brain selects for and creates output and behavior, which is the critical aspect of understanding psychiatric disease in the end. All right, so I'm going to spend the last maybe 10, 15 minutes, 25 minutes, unless they stop me, talking a little bit about some critical things that I learned on the way during my career and how that's informed the science that I do. So as I was finishing up medical school in my MD-PhD, I really didn't have a great sense of like how to make myself useful in the world. I'd spend most of my time in school. I loved the engineering aspects, but I wasn't sure how to really make this stuff matter for the patients I'd seen in the clinic. And I signed up to do a rotation, just a 4-week rotation, seeing a clinical trial for the first time at the National Institute of Mental Health. And I was in the lab of Carlos Zarate, who was studying a new investigational drug called ketamine. and he was testing it out on patients with bipolar depression. And for me, this was like, I'll say at the time, I thought, this is the strangest experiment I've ever seen in my life. Like, ketamine, like, to the best of my knowledge, was either an anesthetic or a street drug that made people hallucinate. And I remember the first time I saw a patient with bipolar disorder get ketamine, and they started describing these weird sort of symptoms where they were sort of seeing things and felt like they were having out-of-body experience. And I was like, yeah, that's what ketamine does. Of course, it's not an antidepressant. And surprisingly, the next day, the patient's mood symptoms had gotten a lot better. Now, fast forward like 10, 12 years, it is an FDA-approved treatment for ketamine or for depression. But for me, it was really cool being there at the early stages and seeing these early experiments and seeing the arc that discovery takes to ultimately get people better and living better lives and more productive lives with their family. So I got fascinated with the idea of translational research and figuring out how to make the things we do in the basic science framework matter. I emailed the director of NIMH, National Institute of Mental Health, Tom Mintel, and much to my surprise, he emailed back in about 45 minutes and then invited me out to lunch. And so we had lunch, and he spent most of the lunch convincing me that I should become a psychiatrist. Most importantly, because no one was going to let me build brain pacemakers hooked to microchips and implant them in people's heads if I wasn't a psychiatrist. And so I gently pushed back, and I said, like, this seems like a great idea. But the problem is, if I start getting trained to be a psychiatrist, I have to stop doing research. And I love doing research, and I don't want to stop doing research for 4 years to become a psychiatrist. And so, he asked me a question that, mentors don't often ask trainees. But he essentially asked me, well, how should the field train someone like you? And it was funny, like no one had ever thought to ask me that question. I don't know why I would be in any reasonable position to answer that. But my response was, well, I sort of stay in the lab and do residency like part-time, like it was a hobby. Now, people spend lots of time going to school and medical school, and then lots of hours in residency to be doctors. And I'm sure most of them don't think of it as a hobby. But I essentially said, I want to be a doctor, but I'm a scientist first, and I want to figure out how to make the doctoring part work of it. And Tom was really fascinated by the model. And before you knew it, he basically created what he said was his N=1 experiment, where he was going to see if it's possible to take a MD-PhD student and turn him into a doctor, essentially training them part-time. And so I started my own lab, and then I did my 4 years of psychiatry residency over 6 years. So I basically did it part-time. And what was so fascinating about this is by the time I was halfway through, I'd so often gone back and forth between being in the hospital in the morning and the lab in the afternoon that I started asking extremely clinically relevant questions. And I realized I would just see things in the clinic that like most of my basic science research colleagues didn't see. I knew exactly where to go. I knew exactly what the relevant question was for improving human health and what needed to be answered. And so I started writing grants, and much to my surprise, right after my 34th birthday, I got my first R1. And this is a really weird thing for multiple reasons. First, as many of you all know, about 1% of the NIH-funded workforce with R1s, about 1% is Black. And then, secondly, for MD-PhDs, the mean time to independence, in other words, the time it takes you to get your first R1, is about 45, right? So I basically somehow this idea that Tom and I came up with over lunch basically totally broke the model of how one would think about training a physician scientist. And the NIH has since funded grants to allow people to do similar experiments with training in residency to see if they could accelerate their time to independence. Okay, so I'll end with a few slides that I think are particularly important. The first one is I'm just putting up a series of pictures from a bunch of the folks that have trained in my lab or are currently training in my lab. And I think it's really important. We do good science. I tend to be a fan of my science. I'm obsessed with, I'll be honest, I'm obsessed with my science. I like it a lot, but of course I'm extremely biased. But if you like the science, I always like to point out that the science we do comes out of a lab that draws broadly on diverse experiences, right? So my lab looks like America, and it is the reason why we're able to fuse ideas and disciplines together. It is not seamless. It is incredibly painful to bring diverse perspectives together, but I think science is much the better for it. And if I talked about each of these people, you'd be astounded, both by how successful they are, right? So there are people here who, some of them were undergrads. I've got a couple of MD, PhD students. I've got a couple of neurosurgeons that are finishing up from WashU, rising neurosurgeons from WashU to UPenn, to Emory. I have students in graduate school now doing their PhDs. I have individuals here who are faculty members. One of them, top row, third from the left, Rainbow, has her lab. She recently opened it in Iowa and recently was awarded the NIH Director's New Innovator Award. So people who just bring an incredibly broad range of expertise and a commitment to diversity in the sciences. And then in the upper left-hand corner, Sherilyn Black, who strangely enough was my first postdoc, but like really wasn't entirely. We trained together in grad school. We decided to do some cool projects together. And then Sherilyn basically decided to somehow, in the course of 10 years, become Associate Vice Provost of Duke. So Sherilyn is like my boss's 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 boss. But fortunately, she still listens to some of the things I have to say occasionally. And what's equally as important is that if you look at this slide, you don't even appreciate all of the diversity here, right? So there are people who are first generation, there are people who experienced challenges with disability along various axes of their training. And we have figured out how to bring all of that as a strength to the enterprise that we built. So you don't get the science without the diverse team working on it. This is a group of some of the mentors that I touched along the way. I'm just going to segue a little bit into some of the fun things that I do outside of the lab that are lab-like. So I'm like all science, I'm all engineer, in my spare time, psychiatrist, and in my other part of my spare time, I'm like all about like advocacy and science communication. So I joined a bunch of my science mentors. You can see Tom Insel on the first row, but also Carl, who you all likely know, Carlos, you can see him at the bottom, Helen Mayberg, and Holly Lisenby, and Patrick Kennedy, a bunch of folks who we came together as part of this PBS series to really help educate the public more broadly about bipolar disorder and what the hope of medicine in helping individuals and families who had been impacted by this brain illness. I also spent a lot of time doing mentoring beyond the work that we do in my lab. So up until the pandemic, I was traveling back to my undergraduate institution once a month to meet with students on campus. I'm happy to come to Puerto Rico. It's a lot longer flight. So if y'all set me up with the hotel, I'm there. but doing mentoring on campus and really trying to help the next generation think through their career plans and their career development as well. So it's something I started on, and I just did it because it was a passion of mine. I'm super altruistic about it. After being there for about a year, I realized it was like the best recruiting strategy ever, right? I'm giving away all of my tricks. It occurred to me that, like, Duke's basketball is great because Coach K tends to recruit, and Alabama football is great because Nick Saban tends to recruit. And I shouldn't just leave the population in my lab left up to who applies and who shows up for interviews. I should bias the application pool by mentoring those many, many years before. And so it turns out this is a great way of building the diverse lab as well. In addition to the advocacy, I also, as I mentioned, I do some science communication and some policy advocacy as well. I joined the director of the National Institutes of Health and the FDA commissioner in 2016. That's us in the upper left-hand corner, really talking about the importance of big data and machine learning in science more broadly, and the importance of creating a more diverse enterprise that capitalizes on all of the talent population here in the US. I later joined the 44th president on a panel where we were talking about precision medicine and the future of neuroscience as supported through the BRAIN Initiative and Innovation and Technology. And as a result of those, I was invited to join the signing of the 21st Century Cures Bill, which provided a good amount of funding to support the BRAIN Initiative as well. I've also spent some time at TEDMED. The TEDMED started off for me as an opportunity to talk more broadly about my science, but it very soon after that morphed into an ability to get training around scientific communication. And I don't think we as scientists often realize how important that type of training is. So I joined the editorial board to learn what was the makeup of a good story and how does one tell a good story. And then I got additional science communication training and spent some time hosting TEDMED. as well, being the opening host a couple of times. This is me in the last TEDMED before the world went a little bit off, and was joined by, or 2 times ago, by the Surgeon General of the United States, who's also graduated from my same undergraduate university as well. And then I was able to spend some time working on a congressionally mandated committee to figure out how to best support the next generation of scientists, and create path and space for them to bring their impact to the field as well. And then finally, the BRAIN Initiative, which I helped author the scientific outlay for the second half. And I'm currently on the multi-council working group as well, helping to steer that initiative forward. Okay, so I'm going to end on this thought experiment. I was invited to give a talk, I guess 3 years ago now, at Abercam. So it's a big conference, like 3,000 people there, diverse background trainees interested in the sciences. And I was asked to give a talk, one of the keynote talks. And this was super weird for me because I'd been sitting in the audience like 3 years before, like, oh my gosh, how do people get so awesome in order to give a talk? So it was really weird for me as I was on the stage. So as I'm preparing for this talk, I started doing this thought experiment and sort of like mapping out like the last 5 years of my career. And I would encourage you all at some point in time to do this thought experiment as well. So I'm sitting there and I imagine myself walking into a big room. I'm all about like I'm a psychiatrist, right? So it's all about like visualization. So I imagine myself walking into a room and there's a big table, right? And around the table, there's 10 people sitting in 10 slots. I walk in, 10 people, 10 slots. And then everyone starts introducing themselves and saying one cool thing about them, right? So the first person goes and they're like, hey, I have an MD-PhD and I'm a psychiatrist. I'm like, that's pretty awesome. And then someone's like, I'm a neuroscientist. I'm like, man, you work on the brain. That's awesome. And then I won a Presidential Early Career Award and I'm an endowed associate professor. And I published this cool thing in Cell. And I was in a movie on PBS and I was a TEDMED speaker and I was opening host at TEDMED. And I was at the conference like with Obama on stage. And I helped author the second half of the BRAIN Initiative. And I literally imagined doing this visual experiment this exercise in my head. And by the time I got to the 10th one, I was like, oh my God, I don't belong in the same room with these people. And it was this exact moment where I realized that I gave myself imposter syndrome. And so this was really informative for me because it occurred to me at that moment that if I gave myself imposter syndrome, it's probably never going to go away. And because of that, it was more just learning to deal with the fact that there are environmental cues that have given me the sense that I don't belong there, but I'm going to be there anyways, right? And so part of the challenge is sometimes we wait to have a series of accomplishments that makes these feelings go away. But here, my accomplishments were making me feel like I didn't belong in the room with myself, right? So I'd encourage you all who have that shared experience of sometimes feeling like you don't belong there, to take a good look at yourself and the success that you've had thus far. If you are in neuroscience, if you're in college, that in and of itself in our society is already something to be celebrated. So I want to celebrate you all, but I also want to help anchor you in the idea that those feelings may remain even after you win your Nobel Prize. All right, so I like to end here. I think this is a particularly important slide. I've talked about my lab, I've talked about me and my experiences. These are my mentors, right? It is not a full slide, it's not complete. There are many more that have been added since I made this slide, but at some point in time, they start getting so small, you can't see their face. And a couple of things that are particularly important. First of all, I'm not sure if anybody noticed this, I'm an African-American male. All of my mentors are not African-American, most of them are not. All of them are not men. All of them are not from African descent. My parents migrated here. All of them are not psychiatrists, right? There are a series of people who we have a shared set of values, and a shared set of values allows us to build a relationship where they drive me forward. I would encourage all of you all, the most important thing that you can do in your career is find mentors and convince them to invest in you. It is very, very, very easy to track my career and predict all of the outcomes and the successes that I've had in my career if you understand these people. I am just a sum of parts of them, right? And this doesn't make me feel bad. I also appreciate that lots of successful people tend to be the sum of lots of successful people, right? So it's nothing to feel bad or ashamed about. That you know, like, we all need help and we need support and we need people to advocate for us. When you get that, stay humble and realize that your success is a function of your hard work in them as well. So thank you all so much for having me here. I went just as long as I promised I would go and not a minute more. And I'm happy to take any questions.
What a great presentation. That's all very, very amazing. Thank you so much, Kafui, for this talk. I am like really impressed, and also for our students in particular to listen to you and give them this these wisdom sound bites. It's so important, in particular for them to address how they're going to go into this career as a scientist and also as a citizen of a very diverse community. So this is something extremely important that our students are very conscientious about. Yeah, I see lots of comments that I enjoy very much the talk. And I meant to ask you, because we have in the public some students that are also in bioengineering, and they have trained in this field, and then now they're trying to enter neuroscience from the engineering focus. What was it that made it successful for you to do that transition? If you can share with them about that.
Yeah, I don't know if you want the good news or the bad news, but it wasn't very successful for a really long time, right? So it's funny, right? Like, there are, like, human bias is such a strong thing, right? And it's exceptional in science because the way we determine what is true is based on, like, our peers, right? So the bias of our peers is generally the thing that gates what is true, right? Mm-hmm. You have to, at a minimum, you have to convince like 3 people that you know what you're talking about, whether it's a grant or publication, right? Yes. And if you tend to think like they do, it's easier to get the truth adopted, right? So for me, a lot of the challenge that I had is just like my conceptual frameworks for things were just different because I was an engineer, right? So it's not like I was like trying to be outside the box. I just was like never in the box. And so I'd sit in scientific talks and read papers. And like, I mean, I would literally at the end conclude, well, that absolutely makes no sense, right? And the things I'm seeing would make no sense to a first-year engineering student. But it's a totally different framework when you're thinking about building a system from, let's say, physics versus biology, right? And the brain is clearly both. So I found that, like, in neuroscience, a lot what was missing was that people hadn't approached the field thinking about first principles from an engineering standpoint. It was thinking about first principles from a biology standpoint. And so my utility was, it was super useful. Like, my strength was that I saw it differently. The challenge was that I saw it differently, and so I had to convince people. This is why the scientific communication is so important. And you also need mentors that sort of help you navigate bringing new ideas and new perspectives in.
That's great. Yeah, definitely. The mentors are so instrumental in this, moving along as fast as you can regarding the environment.
Yeah, but the difference in perspective is the strength, right? That is a scientific advantage. You have to figure out how to overcome the communication, to overcome the bias barriers. And I don't mean bias in a bad way. I mean biases in humans tend to believe what they believe.
Sure. Yeah. Okay. And now, from the science that don't mind this. There's a poll that we have for, I don't know if you're seeing it right now. But the participants can do a poll on the seminar. So
I shouldn't click strongly disagree.
No, you're not allowed to make over. No, just kidding. So the other thing I meant to ask you was how, from the standpoint of psychiatry and your work that you were presenting us about the stimuli, and depending on the level of what are we trying to navigate on the phenotype of depression, how So how do you see your work translated into a therapy or a treatment? Are we talking about that you will incorporate engineering focus on, changing the depressed brain?
Yeah.
Can you talk a little?
Yeah, the simplest conceptual framework to understand what we're trying to achieve. You could appreciate this if you ever have chest pain and go in the hospital. The first thing, one of the first things they will do after they put the oxygen on is read electrical activity from your heart, right?
Okay.
With the EKG. And they are so good at this and they understand these patterns so well that it works on you, even though you've never been in the hospital, they can still read those patterns. So we're essentially trying to figure out what the electrical patterns are. It's not a 12-lead EKG. We have a many-lead brain signature that tells you what brain state you are in emotionally. And then we want to be able to both stratify you to treatment based on that, but also then have a set of treatments like a brain pacemaker that you would say, you're in this state, so then we might be able to stimulate you out of that state using hopefully non-invasive stimulation.
Oh, I see.
Yes.
Now I understand. Okay.
Yeah. So an EKG and a pacemaker.
That is so amazing. And then so is the person, do you foresee the individual manipulating this way of altering the mood in order to him or her, depending on the situation, adapt to it? Or
yeah, I mean, we can imagine a couple of scenarios for this, right? So, the human body is a system that reaches balance, right, or homeostasis. So, you could sort of appreciate the feedback allows you to learn how to train the body to change its homeostasis. Like the Fitbit is a homeostasis changing device, right? So you could imagine if you had a really good understanding of the state that your brain was in, you could regulate it a different way, right? In its simplest form, we as human beings have an ability to do this. If you go to therapy, right, or if you do some mindfulness training, you realize that you have neural systems that can regulate your internal state. And by doing that, you process cues differently, right? The idea is in the case of mental illness, you lose some of that ability to shift your brain states and do the regulation. And so what the device would do was give you back some of that ability to control and regulate your brain into the appropriate state.
Oh, wow. Okay. And would you focus on the medial prefrontal cortex, or I saw that you are also very much interested in the accumbens from the part of the reward and of course, the medial prefrontal cortex for all these aspects of executive function. So
the question is, it depends on the severity of illness and what the ultimate tool is, right?
Okay.
So, and interestingly enough, this is not all 100% in the hands of neuroscientists like myself. As you all know, Elon Musk has a brain machine interface company, right? In which there are probes all, he's basically trying to put probes all throughout the brain. So if the outcome of this is we can do this non-invasively, right? It is indeed possible to stimulate, let's say, the prefrontal cortex non-invasively and cause activity downstream because the prefrontal cortex projects there, right? So just because you have a target area of stimulation doesn't mean that that is the ultimate and sole place in which you will mediate a change or plasticity in the brain. So even if we can learn how things are changing in the interactions between deep and shallow structures or between combinations of deep structures, we can ask the question, are there things we can do on the surface that change those interactions in a deep place? So the more shallow we target, the less likely it is to be invasive, the easier it is to ultimately implement.
Okay. So we have a couple questions now. It says, from Paola Morales, who is one of these students that is a bioengineer major. She says, people have told you you are in different scenarios and compromises. If so, how you deal with credibility and be so diverse?
So no, it's a great question, right? So there is innate human bias, right? And that human bias shows up in two really important ways in science, right? One of them I talked about earlier. This ends up being an advantage if you're different, because the science tends to generate groups of people who think the same way. That's why we have fields, that's why we have camps, right?
Sure.
And so if you're coming from outside, all of your life experiences, your brain is basically a map of all of your life experiences, right? Your genetics and your life experiences. So if you are diverse, you literally have a different set of life experiences. So you're walking in with a different tool to observe, right? And so that is an extreme advantage, but it also brings challenges, because if you are different, then the human bias comes into play, right? So I would encourage you to focus on the idea that your difference brings an incredible advantage in perceiving things in a different way. And then what you want to do is figure out how to have mentors and a community within the scientific enterprise such that your differences can penetrate and ultimately change the way and transform the way the field works, right? This is both a gender topic, it is a racial and ethnicity topic, and it is a field topic, right? And if you sit at the intersection of all those three, you really want to find mentors and guides that help you take that really unique set of perspectives that you have and bring it to the field. You know, you can appreciate this in neuroscience. So I didn't realize this till I got in the psychiatric, the clinical setting, but like 2/3 of the people who get depression are women. And yet the vast majority of animal studies are in male mice. So it makes no sense, right? Like at all. Except it's clear when you think about the bias in the field that existed, and you look 30 years ago and most investigators were men, right? And so they focused in that direction. So it's this incredible gap in bias that has persisted in the field that one might push on that if you're coming from a different set of experiences.
That's right. There's another question. It says, do you have thoughts about how ketamine treats depression? Is there a cardiac analogy that fits?
Yeah, so people have been investigating this. It certainly changes. One of my great colleagues, who I think the world of Connor Liston, has been doing some work looking at prefrontal cortex and how spines, in other words, the connections between cells, are changed by ketamine. Ketamine does look like it's inducing some changes in connections involving the prefrontal cortex, but nobody knows the exact precise mechanism. It's a space that's really rich for investigation and interpretation. You know, if I can be frank, in most of the area of psychiatric illness, like nobody really can pinpoint what we would call the mechanism of what's going on. In fact, we don't even know what level of analysis the mechanism falls on. I'm pretty sure it's not like at the level of electron cloud density, but whether it's like molecules, proteins and molecules, or cellular firing, or network level architecture, we as a field are still exploring this quite broadly.
Great. And then there's another question. I love your talk, says one of my.
And let me say, please, please, please don't disagree that you've met a potential role model. I'd be hurt.
That's right. Quick question. He says, what advice would you give to someone that is about to embark their journey into an MD-PhD? That's Hector. He was accepted to the MD-PhD program with Yale and UPR. So
okay, it's really important, Hector. I hope you hit strongly agree on this question before you started asking for advice. All right, so it's a beautiful journey, and there's no real standard outcome to this, right? MD-PhDs haven't been around for, it's like 60 years old here in the United States, right? Which means the programs haven't been optimized. And if you're from a diverse background, it certainly hasn't been optimized for you. Right. So I would encourage you to find your own way and path through it and learn how to become true to yourself as you're doing it. Right. It's fascinating. Right. At the end of this, people always ask, so like, what are you going to be 80-20 or 20-80? In other words, 80% in the hospital, 20% in the lab, or 20% in the hospital, 80% in the lab. And how are you going to break it up between the two? And I'm like, I don't even know. There are like 5 categories and I don't like. Like apparently there's like science policy and science communication and bench research and translation. Like there are all of these like amazing categories, and you can do a lot of good in each of them. And you can just figure out how to use your skill set to make the world a better place. And learning and training and developing your skills is always a good thing. So focus on that.
That's great. Yeah. And then I think there's a last question. What is your advice to become a communicating leader? Is it possible an undergraduate, since I just started to know about research ecosystem and in the neuroscience area.
Yeah, so if you want to be a good communicator, you got to communicate, right? Like I always say, you can't hit a good forehand in tennis unless you hit a forehand, right? You can't hit a backhand unless you hit a backhand. And you don't have to have the best forehand and backhand to win. You just have to have one of the two and maybe a nice serve, right? So you just gotta figure out what your game is and how you're gonna play your game and tell your story. And you have to develop that, right? So if you're an undergrad, you should already be going into second and third grade classes and talking about neuroscience, right? Like it doesn't require a whole lot of shifts for me to talk to a high school audience as it does to talk to an audience of my colleagues, right? Because there's a sweet communication spot where you can focus on themes and ideas, but you have to practice that. That is absolutely something you can practice right now. It's a skill that not enough scientists develop.
That's great. And I think you forgot one of your skills, which is advocacy for the diverse workforce in the neuroscience, which you have done very well. If you follow Dr. Dzirasa in Twitter, you will know what I'm talking about. And most recently, I just wanted to highlight that he shared this article with me yesterday. And that he just recently published in Science an article about policy forum. It's called Concrete Steps to Diversify the Scientific Workforce. And it's a very important read for everyone that have the chance to see the importance of what we are really doing here at the Endure community, which is creating a real career path for underrepresented students and faculty toward changing, right, this scenario toward an improvement, in my opinion, of making it a community that is really representative of what we all are, right? That is not just pockets of people not communicating and not growing together.
Yeah, can I just make a comment about that? Actually, to the question that was asked before about being a biomedical engineering, bringing the ideas in. I think it's sort of been the same for me on this issue of like diversity and workforce development. Right? Like I've always been doing this stuff, right? So Marguerite and I met in like a program that was sponsored by UNCF Merck for graduates and undergraduates and postdocs, like fellowship training. So I've always been involved in this. It just didn't penetrate back then in the same way that it did now, right? So sometimes when you're doing important and good and critical work, it takes a while for the field to move in a way that they ultimately see the value of that. And in the same way that people ultimately saw the value of the engineering approaches that I was bringing, I think now the entire field has realized that the value of the equity work and the diversity work in making the field draw upon the talent of the population more broadly. So I'd encourage you all, if you're passionate about something and you believe, just keep pushing. Eventually people will get it, right? And now, like my scholarship, if you put up like my publications and what people think I'm an expert in, I'm like just as much neuroscience as I am like advocacy. And each of them are like this own unique, very rewarded, incentivized pillars in my career. But for me, it was just something I believed in, I was cared about. So yeah, push.
Wonderful. Well, I think we are up to the end of our seminar. We really thank Dr. Dzirasa for being here. Kafui, it was a pleasure to meet you, at least virtually. And we always want to be able to invite you again, but you know, you come to Puerto Rico and we'll have a get together, and then our students will have the pleasure to meet you in person. And from the Endure community, we thank you. Dr. Jones London is here, and of course Marguerite is here, and so we thank you for being here. And so next week we have one of our very own, which is Anthony Burgos Robles, who is a young Puerto Rican scientist as an assistant professor of neuroscience at University of Texas San Antonio. And he's now starting his new lab. So we wanted to have the students see, the different levels, right? And Tony is a product of the neuroscience community in Puerto Rico, and now he's his own professor at UT Austin. So thank you everyone, and we'll see each other hopefully next week. Thank you, Kafui, for everything. You take care.
Thanks for having me.
Bye. Thank you.